you doing?
Fine. You?
Good, good.
Okay.
You have a paper for today?
No, no paper. No.
Okay.
Oh.
Let's go. Let's go. Hello.
In the bat cave, as usual.
Hello. Good morning, everyone. Welcome to today's Neuroccino. I'll be the host for today. I'm Valentina Pacella. And we're gonna have one book and one journal. And let's start. I'm giving you the floor, Michel.
Hey, okay, great. Let me share this awesome book. Can you see it? Yeah, all right. This is a fiber pathway of the brain. And as the title indicates, it would be about the brain and about fiber pathways. It's actually one of the best reference in terms of the anatomy of white matter connections in the primate brain. As the author collected in this book 30 years of axonal tracing in macaque monkey, or rhesus monkey, and been able to build an atlas from there that give us some valuable information about the organization of white matter connections. So the book is actually very well done, and I think I spent my entire postdoc working with this reference as I was like exploring the organization of white matter in the human brain. The book starts with a nicely illustrated introduction of the interest of white matter in the brain. I'm going to show you some illustrations. So they start with Vicens here, where you can see like the nice first depiction of the projection pathways in the brain. And they go through all these historical dissections of a white matter pathway. You see like the corpus callosum here, hansenate fasciculus. That would be the forceps major and the forceps minor here, and subcortical area. And you see how people were like not only anatomists, but artists in the way they were describing the anatomy. And there is no doubt that through their work in dissections, there was no fuzziness in the organization of white matter. There was no disorder or mess or whatever. It was like perfectly organized and perfectly aligned together, all these beautiful axons. And again, for a time where there was no MRI, I think it's absolutely fabulous to look at such beautiful reconstructions. Funny enough, you see, like this is a work from Gal, who brought us the phrenology, which is like seriously loved at recently, for a long time, but still is the first who brought the localizationism of function in the brain and the geol parcellation. So he identified and mapped the different part of the brain based on circumvolution and gyri. And although he was wrong in his approach to think that the shape of the brain will deform the outside shape of the skull, he's been doing a fantastic work of anatomy, and you can see here also how he's been drawing and dissecting the brain. It almost looks like an MRI scan. You can see how he also knew that white matter fibers were there connecting different parts of the brain. But I think he was very interested into the projection fibers, linking single areas in the brain with the rest of the body, the brain being the command center. And I think really how you move from localizationist to associationist, because you have those association fibers that you can see here. There are fibers that link region within the same hemisphere together, leading to the concept of networks and function emerging from the interaction between brain regions. This is work of Mayo here. And this goes on and on and on. So this is a work of Desjardins. I know we insist a lot on the German literature, and they did a fantastic job, but French right there as well. And then we start with a methodological part of the book where they explain how they build the atlas of the white matter with axonal tracing. This is a drawing of a monkey brain, and all the black areas that you have are areas that have been injected with a tracer. It's horseradish peroxidase, and so essentially what you're going to see from that is that 20 years of axonal tracing, we didn't inject the entire brain of the rhesus monkey. So there is no knowledge of the entire rhesus monkey connectome. We don't know that. What we know is how those patchy areas are connected in single monkeys in these Frankenstein models that regroup like several monkeys, each monkey is being injected in different areas. That's what we know. And this is our gold standard. And this is tremendous work, but just give you how little we know about like the organization of white matter connections. So each case will be further represented in the book. We can assess exactly the connection and the connectome of those regions. And then you have like composite maps that regroup all the cases together, where you can have like a more global view. Quick word about the cytoarchitecture. And then they show like how with the tracing, with Nissl stain, they can actually differentiate the existence of the fronto-occipital fasciculus from the corpus callosum and the corona radiata, because here you can see the organization of the white matter this way, and here you see it's antero-posterior, right above the caudate nucleus, which is completely different from the cingulum. A little bit of anatomy, because when you write an atlas, you can go straight into like, hey, we're going to talk to you about white matter, tell you about all this region connects, without like having a quick reminder of what are those regions. So you have this nice depiction of the organization of the surface of the monkey brain in terms of sulci, but regions. They're mostly characterized by the equivalent of Brodmann cytoarchitectonic areas. They have those weird names: TS1, TS2, TS3, T1, T2, T3, TEO. You know, it's a mix and match between cytoarchitecture, functional field, anatomical areas. Yeah. And this will be all the slides I will be studying in the atlas. So it's been like nicely cut in many slides. So you have the anatomical sections here. You can go through this monkey brain. And then we'll start showing you the concept of the axonal tracing. So, so they inject over here and they let the tracing travels through the white matter for weeks. And then with special cameras, they're able to take pictures, and that shows you where the tracer has been going through those axons from the injection site. So you can see that the further you go, the least visible is a tracer, which is not only representative of like a dilution of the tracer, but also the number, like the further you go, the least axon you will have connected to the region you're speaking about. So there will be also like a kind of how we can divide like the different kind of fibers. You have short U-shaped fibers, neighbor association fibers, striatal fibers. Commercial fibers are going to go to the corpus callosum mainly, then thalamic fibers and ponting fibers. This will go to the subcortical, more striatal fibers. and so on and so forth. And they're all mixed up. But the advantage with the axonal tracing is that you don't have all the limitation of neuroimaging. And then we start with, remember all the numbers that I gave to you for the site of injection. We have for each of those cases, a full depiction of what is connected. And so you start with this case with like this little injection here, and you have this atlas that show you exactly where those connections are going, how they are organized from this site of injection. You see, like it's a hot mess and you get like a lot of different things, but the level of precision and the clarity of the findings is just amazing. And it's still the case number one, you're going posteriorly, and you have like the real picture over here, that, you know, because this is a drawing. And then you have the real picture over here that show you like really the raw data from which the authors have been working on. And then it carries on with case number 2, case number 3, case number 4, case number 5. And then you have like those composite maps, 1 point, which will be case 1, 2, 3, 4, 5, and 6 that are regrouped because they're all part of the parietal lobe of the rhesus monkey, which is very small compared to the human. And so they color coded, as you can see here, and following the color, if you're not colorblind, you can see how those different areas have a topography inside the brain and project in different portions of subcortical anatomy and other brain regions. I have to say that some of those slides, you know, start getting a little busy, and this is just for those tiny injections, and they give you a little feeling on how complex is the organization of the white matter. So look at this. It's like, oh my God, this is the entire white matter with all these 6 sites of injection that is currently involved. And then we carry on, and this is a new chapter, this time on the superior temporal region. Same thing, you have all the case. And then you have the summary map right here of all those cases of the superior temporal. And you can see again how complex the anatomy is, but I really feel, you know, it's a unique window of understanding into the connections, and the fact that it is complex is just, you know, there's nothing we can do about that. And then the inferior temporal region. It's coming up. There you go, inferior temporal region. Then again, you have all the organization here. And then, what's the case 17 about? And case 18. Oh, I think we're starting with the occipital here. So that's the entire part of the occipital lobe. It's coming. And you see that like dorsally, you have those projections to the frontal lobe from the occipital lobe, but you have no projection from the ventral region, which will be the inferior frontal occipital fasciculus. Having said that, the atlas does not have medial injection because it's really hard to do. No, it has, I think, B1 injection. So, but if you were to think that the inferior frontal occipital fasciculus project from advanced visual areas to the frontal lobes, then there is none in the macaque monkey, at least not with axonal tracing. And then you have the cingulate, gyrus, or limbic system, same thing. Goes for 673 pages. And you see here, and we start with the frontal lobe. So that will be the organization of the motor cortex and all its connections. Where is it going? And then I think then will be the prefrontal cortex. No, there will be medial cortex. And then that's the dorsolateral cortex. Coming, orbitofrontal cortex, and the summary map is coming here. You see all those fibers doing like this, reaching at the top, and at the frontal ascending tract. So it was not described in the book, but I could validate by going through the book and comparing with human dissections. And then the authors tried to summarize the anatomy of the white matter connections by putting together all the slides and doing this kind of 3D representation of how those tracks should look like. So you got the SLF1, and this is based on all those slides. So you got the shape over here, so going through coronally, and that allows him to reconstruct this. And then you have the SLF2, same story again. SLF3, arcuate fasciculus, and every time you have all the reference and all the works that have been associated with these tracks. External capsule, that would be the middle longinal fasciculus. Uncinate fasciculus. Cingulum bundle. And inferior longinal fasciculus. And then you have like some advanced analysis that are coming up. Fronto-occipital fasciculus is coming next. Whereby they try to combine the information from different regions and project it onto track of interest, such as the corpus callosum. And you can see here how different points in the brain project in different area of the corpus callosum, and that's a work from Pandya and Rosine in 1985, where they divided the corpus callosum according to the projection from the cortex, and they try to reproduce this based on all their cases and the entire atlas, and this is a result that they have. And so you know that in the corpus callosum you receive afferences from these different regions. Here, first color map of the midsection of the corpus callosum. They did the same thing with the internal capsule that you have here. So you have the organization of the internal capsule according to its projections. And then the sagittal stratum, again, same idea. And then we finish with the full brain with the white matter divided according to the projections of the white matter tracks. To be honest, I have difficulty to read those maps, but it's still very informative. The end of the book is all about neurology and brain pathology of the white matter and interpretation and the link between white matter and syndromes in clinical neuroscience, including you just saw it. Where is it? Visual neglect here. The work of people on visual neglect. And that's it. And then billions of references you should all know. And end of the book. Questions?
Thank you, Michel. Any questions? Go ahead.
I do have one. So I wanted to know, I have two questions. First, this work that is incredible, it's quite old now. I think it's about 15 years old.
Mm-hmm.
And was there any updates on this? Because I imagine that the research continued with the tracing of a monkey's brain.
Yeah. Well, how do I say that? Not so much. Mainly because tracing is really you gotta sacrifice a monkey. But as far as I know, in terms of updates that were just on the neuroanatomy, Pandya published a paper about 5-6 years ago. And let me show you where is that brain with all the injections. I lost it. Where he's been injecting really early connections of. He's been looking for the inferior fronto-occipital fasciculus, and they didn't really find projections by injecting the early visual areas. So it's still a work in progress. And then most people they don't really do huge white matter pathways like this. So research that people are not doing anymore. Now they're doing like micro systems and micro connection like Kathleen Rockland. So yeah, there's not been like so much progress. And again, this is a lifetime of the team of Deepak Pandya, who was the first, or one of the first, to actually do that. And he's been training Jeremy Schmahmann, Mike Petrides, who are carrying on, and Kathleen Rockland, and Yetarian, who are carrying on the job, but on the field of interest, which would be more cyto architecture, macro connectivity. I don't know what the Italian is doing. He's been doing these latest papers 5 years ago. And Jeremy Schmahmann is now he did his book, and now he's a famous director of an institute. So I don't think he's doing external tracing anymore. So yeah.
Okay, thank you. And my second question was, how comparable is the macaque brain or the monkey brain in general to the human brain? I assume that it's not a one-to-one correspondence, but how much of equivalent areas and tracts can you have between the two kind of species?
It's a very good question. And so it really depends on the authors, and like people working on the macaque brain will be like, oh yes, this is very comparable to the human brain, yet it is quite different. And you can see it just when you look at the surface anatomy. Where are you? There you go. So so see, for example, here, this is the orbitofrontal cortex. We can't see it really in humans because our inferior frontal gyre is going to go up to here and cover it up. And then like the frontal lobe of macaques, is divided in two. superior and inferior. You don't have a middle frontal gyrus, and you don't have a middle temporal gyrus. So those are big difference. Then this is a parietal lobe. This is so small in proportion compared to humans. We always speak about disproportional extension of the frontal lobe in human compared to macaque, but what about the parietal lobe? And then I think like the thing that is like the carriers that I can see is like you can see part of the calcarean fissure on the lateral part of the brain, which literally means that if you speak about evolution, the primary visual cortex was here, and then the parietal lobe has been extending and pushing it into the medial section. And this change, of course, with it, the anatomy of white matter connections by dragging it with it. And that makes like big difference. And then I guess if that's like the gross difference, when you just look at the brain macroscopically, if you look, with a microscope and you look very closely, there must be major differences as well. But, they will require having comparable measures in all species, which, we're not going to sacrifice human and do external tracing. And then another dimension, the shame Steph is not here, is that people are different, and macaque monkeys are different. We did a paper on this in 2017. The level of anatomical variability is actually highly comparable between the two species, and you have different phenotypes, different shape of the brain, you probably have different white matter connections, and this is going with different functioning of the brain.
I'm gonna jump into this question. And I wanted to ask you, and what about the lesions or disconnections? Are they comparable between monkeys and humans specimen?
Yeah, so the literature on disconnection is interesting. So if you cut the uncinate fasciculus in macaque monkey, you have a deficit in memory and associating like emotion with episodic memory. And HM got a disconnection of the uncinate fasciculus and had a lot of episodic memory impairment. He also had other disconnections. In macaque monkey, if you disconnect the frontal lobe from the parietal lobe, they will also show visual neglect. There's been a paper of Gaffan in 1997 about this, showing this kind of effects that are comparable to the human. Although at that time, Gaffan was testing this hypothesis, and we didn't know that in humans that was the same, and then we showed that in human it was the same. But I think like the most interesting, and why people have been using a lot the disconnection and the brain damage and lesion in macaque monkey is a Parkinson model, because you can actually mimic, Parkinson behavior and work on to trying to compensate for symptoms using the macaque monkey. And like the translatability from macaque monkey to humans is pretty good. It's just very expensive, and ethically you cannot run experiments on 100 monkeys to be sure that they're okay. So people do it in mice, and that's why we've been trying to push for like trying to find a species in between that will help like the translatability while still being usable in high numbers, to be sure that what we're doing works.
Thank you very much. Really interesting. Any other question before we move on? Okay, I don't see any. So thank you, Michelle, for the book presentation. And I'm gonna present myself a paper on share screen on a very famous author, Italian author, but I think you all know him. and it's Dante Alighieri. You may be familiar with this masterpiece, the Divine Comedy, and La Divina Commedia. And this is the 700th centenary of his death. And so in Italy, we have many celebrations and talks about his work. And it's been recently published, this paper on Dante, on mind and the brain, on cortex. And I wanted to give you an overview of this nice chapter. So the theory of mind and brain that Dante talks about into his works, they are not original. So he is basically a mirror of his time. And the paper starts describing how Dante conceptualized memory. And he uses this nice metaphor as the book of memory, because according to his time, memory was comparable to a book where in the first stage, memories are encrypted into the mind, but they are not accessible. They're not accessible to the intellect, and then they are transferred to a more stable stage where they can be manipulated by the intellectum. And then there is the concept of anima and mens and spiritus. And in particular, I think that it's really interesting how he describes that body and mind communicate via the spiritus of life. It's very complex. Its philosophy comes from the Aristotle hypothesis on how the mind work, and in particular, according to Aristotle, it is divided into three parts. There is the natural spirit that is starts from the liver, and it passes the vital substances like food to the body via the venous system. Then there is the vital spirit that starts from the left side of the heart and travels throughout the arterial system to reach the brain. And then there is the animal spirit that is the basis of and regulation of body movements and mental processes that are carried out within the brain. Then the piece goes forward talking about sensus and intellectus, because it's always from the Aristotle theories. We have three kind of the soul is divided in three types, in vegetative, sensory, and rational, that Dante describes as live, feel, and reason. And there are three main rules according how the mind and the soul works. In particular, there is the rule that everyone has a soul, especially also animal that has a kind of lower function of the soul, and human have the whole three function, and it describes them as perfect, because they have the whole soul, and it is written here yeah, it's the more perfect of all. And finally, the mind relies on the reason part of the soul. And then the author of the journal, of the paper, describes how Dante consider virtue and passion that are within that are part of the mind and it's part of the human nature, and in particular a very hot topic in Dante's work. It's very famous how he considers love as a duality between virtue, that is the moral virtue and passion. Finally, the most interesting part, I think, for us, it's how the mind works. And I think, well, of course, we can see some of the current knowledge of how the mind works also in Dante's work, because it comes from Aristoteles and Nemesius inscriptions. And so the mind is divided in between internal senses and intellect. Internal senses describes how we read the external information via the five senses, and then we combine the information of the five senses into a whole, with the in a stage that is called a common sensation. And then there is the imagination stage, where there is the storage of the information as a whole about an object. And then there is the fantasy or fantasia stage, where we can manipulate this information of the object. Then there is the cogitativa stage, according to which we give emotional and affection balance to the image of the object. And finally, there's memory, which is a stage where the memory of the object, the information of the object, are stored and can be manipulated by the intellectum. And the intellect is, I think it can be referred to as our modern concept of cognition, because it's separated from these internal senses, and it can manipulate all the information at the various stages of the internal senses in top-down way. It cannot be modified or influenced by the senses and the external information. So it is not, according to Dante's time, cognition wasn't bottom up. And finally, there is a brief this anatomical description of the brain that resembled the functioning of the mind, and it is very primitive, very ancient, because Dante's view was relying on Nemesis, which is an author of the 390 century before Christ, and according to which the brain is divided into three ventricles or cavities, where the psychological processes happen. And these three cavities are the anterior one, where sensation is processed, the middle or central, where the thought happen, and finally a posterior one, or cerebellum, where's the memory processes. To these cavities and to the lesion of these cavities correspond a specific impairment to these three processes. And so Dante relies on this information for the work. And that's it. Any questions? Okay, I see no question. Oh, yeah. Okay, go ahead.
So this ventricular theory, did they think about how the ventricles were exchanging together, or in any way, like a kind of connectivity, ventricle connectivity, or or
I don't know, actually. Here it's not doesn't refer to any kind of communication, but for instance, when he describes the lesions to the three ventricles, they seem to be very concrete, very independent of one another, because if the sensation part is lesioned, a person cannot, the five senses don't work, but one can still reason and have memories of his life. And it's the same if the thought is lesioned, then you can have the five senses, you can have memory, but you cannot process or manipulate them, you cannot reason. And again, memory, if memory is impaired, you don't have memory, but you still can reason on the information that are coming from your five senses. And if the three of them are impaired, the whole creature being in danger or of perishing. So anatomically, it doesn't say if there is a connection, but for sure there is not cognitively, according to the processes. Yeah, Michelle.
Got another question. So, because like, you can have those dissociation in the symptom shows. Would you say it is a first description of what a double dissociation is and how we can segregate function based on the mechanism of studying double dissociations?
Yeah, that's what I was thinking reading the piece. It's like the first description of localization of cognition. Yeah, it strikes me actually. All right. I don't see any other questions, so I think we're okay for today. Thank you so much for joining the Neuroccino, and see you next Monday with Dr. Stephanie Folker. She'll be back to her position, and see you next Monday. If you have any journal, any paper that excites you, or a book that you find interesting, come here, let's discuss it together and have a cup of coffee together. Bye.
Thank you. Bye.
Every day I fit my brain.